Very good. Can you start? Okay, so let's go to the second part, which is the inverse folding process. So we start with an idea, a function of a protein. We were able to get a structure to that function. And now the idea is to get a sequence that you fold to this structure. There are a lot of ways to do this, but we explore a way that is based on graphs. So here I have a protein in the 3D space, and this protein can be represented by a graph, right? So we have nodes here, and we have connections. We have adds between those nodes, right? A node can represent, for instance, a position in the protein. This one, for instance, is a node that represents alanine, a specific alanine in the structure. We can just have this descriptor. I mean, it's just an alanine. But probably, let's go to this one that's better, this threonine. We also have a threonine here, right? It's the same residue. But this threonine here is surrounded by those residues here, alanine, arginine, valine, and serine. And this one is just surrounded by this valine. So actually, it's not the same residue, right? It's the same amino acid, but if you are able to take a representation, those representations should be different, right? So each node can have an attribute that can describe, for instance, the amino acid or characteristic of the amino acid that are in this position. And the adds usually are distance, right? Euclidean distance. So for instance, you can add an edge between two residues if those residues are close enough or are in the distance lower than below 10 angstroms, for instance. So by doing this, you start with structure and generate your graph. But maybe your problem is, I don't know which is this residue. Suppose that you don't have a tag here, you don't have the name, the letter T here. And you want to predict what is the residue based on the environment, right? What you can do? You can, for instance, train a model to make this prediction. And if you predict the amino acid disposition, you are basically doing the inverse folding, right? If you, for instance, remove the tags for all the nodes, this is what the inverse problem is. You are trying to predict the amino acids in each node, in each position of your protein, right? One very nice, relevant, important article is the article of Tom Giacola that proposes a graph-based method for design proteins, or the inverse folding. His method has two parts, the structure encoder and the sequence decoder, right? You can see the encoder as taking the maximum number of information that you have from a structure and create enriched representation of nodes and adds, right? Each node, for instance, can be represented by a vector of numbers. Those numbers, for instance, can differentiate these two threonines here, right? with the information generated by encoder, you can perform the decoding process. I mean, this is a node. Actually, this is not a graph representation, right? But suppose this is like kind of graph representation. You have like, this is like the polypeptide chain of the protein. So suppose you have this position here. The decoder will predict which amino acid is in this position here. So the model predicts this a methionine, then a serine, then the glycine, then the isoleucine, then the alanine. And now we want to predict which is this residue here, right? One thing that I forgot to say is that in the encoder information is basically based on the neighborhoods of those residues, right? So if you want to create a representation of those nodes here, the nodes that are at a given distance from this node here, you'll be contributing to this representation, right? In this case, all these blue arrows here are residues that are contributing to the representation of this residue. Now in the encoder, the decoder, we still have the contribution of the residues that are close to this target residue. But we have additional information, which is the sequence. So now we know that those nodes here are composed by serine, valine, alanine, and isoleucine. And this is used for the model to predict which is this residue in the decoder process. The decoder process is autoregressive. So you predict each node, each amino acid at a given time, at a given position, one by one. And you can see what you already predicted, right? This is important. Okay, a classical example that we delve deeper on this is the protein NPN. Protein NPN was based on this article here and does exactly the same thing, trying to predict residues, the amino acid based on a structure, a backbone structure. So, in protein NPN, we still have encoder and a decoder. So, the information important for the encoder is just backbone distance. And additionally to the backbone distance or representations, in the decoder we can see sequences. And the sequence guide the process of generate new sequence. In a very high level, each protein used as input are described by representations of nodes and adds, right? The representation of nodes are just zeros in the beginning because we don't have the information about the amino acids in the backbone structure. We just have the information of the distance between residues in the protein. And we encode this information here by just using distance between backbone atoms. These representations here are just arrays of numbers, are submitted to the encoder. The encoder generates new adds and new representation of nodes that will be used based on a given sequence or part of the sequence that were already decoded to generate and output the target sequence to fold into this structure. I talked about later this, but the decoder here in protein NPN is not autoregressively because it decodes the sequence randomly. So you don't need to start with this residue here. You can start with this one, then go to this one, then go to this one, until you have filled it all and predict all the nodes of your protein. Okay, so we try to explain here the 3 main steps of this method that can be generalized to other methods or optimization of methods. So the first process, which is common in machine learning, deep learning methods, is the featureization process. So you have your structure. You need to convert this in other things that you can use for training a model. So we extract from this structure, the backbone structure, 3D coordinates, chain IDs, residue index. And this function here detects and identifies the neighborhoods at the maximum of 48 of each residue here, right? So if you have, for instance, 100 amino acids in your protein, you'd be detecting 48 neighborhoods of your 100 residues, right? At maximum. So we end up with an array like this. We have your nodes now, your positions, and the neighborhoods of each position. This method needs to be like equivalent to rotation. So if you rotate your protein, doesn't matter. And a way to do this, very simple way, is to compute the distance between the backbone atoms of each residue. So these are edges, because you have a residue here, another residue here, and you are computing distance between those residues. So these are the edges, right? So you have the nodes with the residues, and the distance are the edges. Okay, so for each pair of residues, each pair of nodes now, we have distance. They include also the oxygen and a virtual C beta. So for each pair of residue, you have 25 distance, right? But it's not appropriated to use directly the distance, right? So 5 angstroms, 10 angstroms. Actually, this is training on crystal structures. So some positions, you are not sure about some positions, right? So you need a way to make a better calibration of this. So they decide to use radial Bayesian function. It's very common in this case. So basically, considering, for instance, a distance of 20 angstroms, you build some kind of bins. And for a given distance, for instance, the distance between this nitrogen, this carbon here, it's 8. No, yeah. 4, yeah, it's hard to see. 4. So you'll be basically building a Gaussian center at this value of distance, right? And if you separate this in 16 bins, now you don't have anymore 25 values that corresponds to your distance, but you need to multiply this by 16 positions to get your full distribution. So by doing this, what we have is 400 values that you represent the distance between 2 nodes, right? So now the nodes are characterized by this 400 vector. It's important to know also about the position of each node in the primary sequence. So they just add also positional encoder, a positional embedding here to make sure that you know that, okay, this position, this amino acid is the first position of your protein, the other the second, and so on. So now you have array, and each pair of nodes are described by this vector here with the size. This is submitted to a multi-layer perceptron, neural network, that will generate this new representation. This is a trained representation now with this size, with this length, 128. These are the edge features, edge E, right? The node features have the same length, but they just have the neighborhoods, for instance, in rows, and the nodes in rows, and the representation of each nodes, the features in columns. But we don't have features here. All the values are 0 because you don't know what is the residue at each position, right? You just have backbones, atoms. We don't know which residue is there. Okay, so now we have initial representation of nodes here with all of them zeros, and a representation of the edges. This will be submitted to the encoder, and both representations will be updated, right? It's an enriched representation. You see why this is important. So let's do step-by-step the encoder here. Okay, you have representations of edges, you have representations of nodes. If you want that the information flows between the edge representations and the nodes, you can concatenate them, these vectors. So you concatenate this, and you create this new array here. It's called edge EV. Why is multiplied by 3? Because you have the representation of, for each pair, right? You have the representation of the first node in the pair, the representation of the second node in the pair, and the representation of the edge. So in this vector you basically show you all the features that you need, the features of the nodes and the features of the edges. If you submit this into MLP, basically in PyTorch, a linear layer followed by activation function, you obtain a new array with the length of 128. This is called edge message, because when you do this neural network here, you are combining information. There is a flow of information between the representation of the nodes and the representation of the edges. This is important for the model. And this is the name, actually, of ProteinPen, is message passing neural network. So by doing this, you can obtain a first representation of your edges, right? If you take all the representations of all the neighborhoods of a given target node and sum all of them, you reduce the dimensionality of this object here, right? So now you have all nodes represented by a feature of 128. This step is called like aggregation. You're just perform a summation over the neighborhoods, right? So let me see. I think I can go back. It's better. So suppose you have this residue here, and you have the neighborhoods. You have the representation of the neighborhoods, right? The summation, you basically aggregate the features of all those neighborhoods into this node here. So now, if you take a look in this HV here, the HV, you know the representation. In this object here, we are integrating the representation of the neighborhoods and the features of the edges too. So this is very enriched in terms of information. You just do a more step of a neural network, this time a position feed forward-wise layer, because you have just the features of the nodes. And while you're doing just this, here it's just updating these features per each node. And then we end up with HV, that is the updated features of each node, right? You can do this in multiple steps, so this encoded here will be concatenated again, and now, you remember that you start with HV with just zeros, but now in the second step, we have a good information for these nodes to be concatenated to the HE, right? And then you do the same steps, and you just keep this HE and the HV, right? In both cases, you have a rich representation of edges and nodes. Okay, I mentioned that the decoding also see some parts of the sequence. So you extract, if you have, for instance, your input sequence, convert this, you create like an embedding. So each amino acid of the sequence will be represented by a vector of numbers with the length 128. This is the HCS. You'll be concatenated the information of the sequence with the information of the edges and the information of the nodes into a single array called HESV. In parallel, you perform the same concatenation, but this time just using HS with zeros. Because in this part here, we don't want to see the sequence. And in this arrays here, you can see the axis just to say, okay, we don't see the sequence here. You perform 2 steps of masking. It's a backward masking when you have the sequence and a forward masking when you don't have the sequence. And sum them to obtain a new HSV array, enriched. The reason to do this masking is because when you are doing the decoding, you just want to see the sequence that were already decoded. You don't want to see the sequence that you did not decode yet. You haven't decoded yet, okay? As I said, the decoding order is not anymore as classical approach from the left to the right. It is random, right? Yeah, here. Yeah, so you take these arrays, you input this into a decoder to generate the probabilities of each position that you are trying to decode, right? I'm not going to detail on this because it's basically the same thing. But at the end, you have neural network, MLP, that will return logits, 21 in this case, because you have the amino acids 20 plus X, which is unknown amino acid. This will go to a soft mask, will return the probabilities. So you have, like, for each possible position, the probability of each residue, right? By doing this, you can, in the sampling, generate new sequence. One important thing here is the sampling temperature. The temperature is a value that you divide the logits. So if you have, for instance, distribution, if this temperature is very high, you obtain, for instance, a uniform distribution. So you can basically randomly pick any of the amino acids. But the temperature is very low. Your distribution will be, you look at the distribution, will be more shallow, and you basically be more confident about your prediction. If you increase this temperature, you generate more variability in your designs, right? And if you decrease this, you reduce the variability in the sequence generated. And how can you test if these methods can really work, right? We just recently performed a benchmarking using some of those methods. So you are interested in designing TCRs, right? As I said, the TCR is called TCR receptor. It's a receptor of a T cell, and it's composed by 2 chains. And those chains can bind to antigenic peptides, tumor-related peptides, for instance, that is exposed by MHC in antigen-presenting cells. So if you can, for instance, design a new TCR that can recognize specific peptides bound to this MHC, this TCR can guide your T cell to, for instance, tumor cells and destroy them. But TCR is not a simple system. They come from a somatic recombination. It's composed by many loops. We have actually 3 loops per each chain. And naturally, this complex doesn't have a high affinity. So to test the protein design models, especially the last one that we'll talk about, the graph-based methods, we have input backbone complex. So this is an initial complex, can be, for instance, a natural interface. We select some residues that are in contact with the peptide and MHC, and we try to redesign these positions, right? The method doesn't know which is this residue here. It will not consider this. But it can, for instance, consider the peptide at amino acids, the MHC, and the other amino acids in the TCR, right? But the one that are the target, for instance, these red ones, it will not consider, right? So the idea is to redesign those interfaces to see if you can obtain a better TCR. We tested different methods, including protein PNN, ligand and PNN. As I mentioned to you, protein PNN just work with backbones, but ligand PNN can include the the side chain contests, which could potentially help you in the design, but we tested this. And ESM. ESM is another method that is used basically a graph neural network. It was not trained on multimers, I mean, 2 assembles of proteins, just monomers. But even so, it's able to predict with a good accuracy the residues there. And Rosetta Fold. Rosetta Design. Rosetta Design is basically, we are using as a baseline, considering that this is a only physics-based approach. So we test those methods, we benchmark those methods in different design scenarios. The one that I will mention here, and we explore more in this article, was the design of just the CDR3. It's basically a loop that is more in contact with your target peptide in both chains. Okay, but to test those methods, we need to have reference, right? So those methods are based on structures, right? So you need to have complex of the TCR with the peptide and MHC to see if these methods can work. So we build our data set of TCR, PMHC. PMHC is, when I say PMHC, is just the peptide with the MHC, right? We create a data set of natural TCRs that are available in the PDB. So the idea is to use this as initial structures for the redesign and compare our designs with these natural TCRs. We hope with this also to capture some kind of diversity because you have different bind modes in the TCR, of the TCR. This is a first result. Usually in protein design, one main or first metric that we use is compare the design sequence with the natural sequence, right? You just compare how many residues, amino acids, you could recover in comparison to the wild type, right? So if you have, for instance, 100% here, it means that your redesigned sequence is exactly the same as the natural sequence that you are using for the design, right? What you can see here is that, here are the test sets that you use, the reference complex, right, 32. And for each of those cases, we're trying to design 10 designs, which are represented here by those circles here. In the horizontal lines, we have the average over all the test cases, right? What we can see here is that protein PNN, ESCM, and ligand PNN can perform better in terms of sequence recovery. I mean, they are recovering more amino acids in comparison to the Rosetta design, which is this one here. But there is another thing that is very important to note here, is the variability, right? We have, for instance, some test case in which the Rosetta design, just based on physical principles, are better in terms of sequence recovery than methods based on neural networks. We did not observe any improvement in terms of sequence recovery if we compare ligand PNN with protein PNN. Just some case here like this one here. But in general, it's basically the same. So in our tests, providing side chain information for the design does not help in the sequence recovery. I will not explore here, but we perform a lot of experiments trying to understand what are the factors that affect the sequence recovery, and why some methods, for instance, this case here, for this test case, we have a high sequence recovery from protein PNN, but a very low sequence recovery with, a very high sequence recovery for ESCM, but a very poor low sequence recovery for protein PNN. Why this happens? We try to access this, but you can, maybe you are thinking, okay, but those methods are training on structures in the PDB, right? So we'll be expected, for instance, if the models see a backbone, I'm just trying to propose glycines, because glycines are high frequent in loops, right? Another hypothesis is that, okay, the models can just be memorized the sequence from the PDB. The models can just consider contacts not with the PMHC, but with other chains of the TCR. So we did a very nice test that is, let's just remove the PMHC and try to redesign the same way. When you do this, we see that for most of the case, in both methods, protein PNN or ECM, we reduce a lot of the sequence recovery. There are some cases, like this one here, that doesn't matter if you have the peptide MHC or not, the sequence recovery is the same. Doesn't mean that the sequence is the same, but the recovery is the same, right? We actually were not able to find the reason for this. Okay, but doesn't matter if you just try to design a protein and you have a high sequence to recover. Actually, sometimes what you want, it's a very different sequence, right? You may want variability. So even if you have sequence with low sequence recovery, those sequences can be good binders. So the idea was to, okay, so let's try to model all of our designs. Remember, the score confidence. We try to model all designs and try to extract the confidence scores for each design to see if the confidence can say something. And here is for some test case. Each circle here is a design. Here's the horizontal line represent a model confidence of the same model confidence of the TCR model that I show you of 0.85. So designs that have the confidence higher than this can potentially be good TCRs for that target. We cannot just, because sometimes this is like prediction, the modeling with AlphaFold, right? So, but sometimes you have a good confidence, but when you remodel your design, the orientation of the TCR is totally different. The orientation of the binder is totally different. The contexts are totally different. So you need to evaluate also the deviation, especially in those loops from the native structure. In most of the cases, you don't have higher deviation. This is an example of a design superposed with the native structure, which obtain a very good confidence. Okay, but it's still important to know the affinity. Okay, we have the model. We probably the model confidence say that you probably have a good TCR designed, but what about the affinity of this TCR to the target? We don't have a wet lab yet, so as I said, it's important to have more steps of in silico validation before to go to the wet lab. So we can prioritize some designs before to go to the wet lab. Because sometimes your collaborator just can test 3 designs, 10 designs, and if they test the first 3 designs and doesn't work, they will not try the others. So it's important to prioritize designs in order to test better hits. So we decided to, for each model here, actually for 7 test cases, or just 7 test cases. We decided to, based on the models, perform molecular dynamic simulations. And then use MAP-PBSA to estimate the binding free energy. But as I said, the system is very special. It's not like conventional protein-protein interface, or it's not related to the interface of antibodies. Antibodies have a higher affinity. But here I'm looking at most of the case low affinity interface. So before to just make the computation, the calculation, and say, okay, you are improving the energy or not, we decided to benchmark the MD simulations with MPBSA for those cases. But for this, you need to build data set of structures, TCRs bound to structures, with experimental affinities. And you did this. We don't have many solved 3D structures of complex between TCR and PMHC with the corresponding binding energy estimated experimentally. So we just have, for instance, this is in terms of delta delta G, but we just had like 7 Ks. But if you compare the experimental delta delta G with the our calculated delta delta G, we see a very nice correlation. Actually, the referee of this publication asked about the modeling. Okay, you are comparing in terms of X-ray structures. So, sorry, I forgot to mention about this. We have crystal structures of the native structure of the TCR and some variation of this TCR, a mutation with some mutations. So we have the wild type and the mutated TCR, right? Both with crystal structures and both with experimental binding affinity measurements. And then you compare, you try to correlate with our calculator. We use the same mutations and do the calculation. But how is the correlation if you, instead of use crystal structures, use models of the mutants? So we consider that you don't have crystal structures of the mutants, and we try just to, base it on the wild type, just mutate the residues of the mutation. We can expand our data set, and we observe that the correlation drops a bit, but it's still useful for us, especially if you try to determine some cutoff to say, okay, I can predict with accuracy of 0.85 when you have an increase in delta delta G of 2 kilocalories per mole, for instance. So despite the correlation is not great as the X-ray structures, we're still confident that you can check if your designs are better or not than the wild type. And this is what we did. So these are 7 test cases. Designs with protein PNA here, and designs with ESM here in red. The calculated delta G for the native complex are in green in both cases, and all the other box plots are the designs. What you can see is that in most of the test cases, we don't have any significant change between the designs and the wild type in terms of binding affinity prediction. This is good because at least the designs are not worse than the wild type. It means that the method is not improving or optimizing the interface, but the proposed residues are not bad for the interface, right? You are creating diversity. There are some cases, I forgot the arrows, but I think it's this one here, for instance. Another one in some place. There are just 3 cases that we identified with energies calculated delta, estimated by the calculated delta G, better than the wild type. So our impression is that those methods are not directly optimizing the interface, but they can be used in pipelines for optimization if you want to optimize this interface. But 2 cases that are, sorry? Oh, yeah. Yeah. Replicas. It's not too much. For each replica is 4 nanoseconds, so you have a total of 60 nanoseconds. But we increase the time of simulation and did not improve. So we decided, actually there is an article, an old article that tried to do this, so you basically consider that increasing the time of simulation didn't improve our predictions. So to do this for main designs, you just keep a short simulation. No. It's a fixed backbone method. So you have your fixed CDRs. You cannot increase the length of the CDR, and you just design using this initial state. But it would be nice. You can use, for instance, people are using, for instance, for antibodies, diffusion models to generate CDRs with different lengths, for instance. Yeah, but there are two cases, 7PB and 7PW, that protein pinning or ESM were not capable to achieve a better or even a similar energy than the wild type. So we try to identify why this is happening. So this is the interface of the first case, the 7PB. You can see there is arginine here that makes this good contact with this aspartic residue. But the rotamer of this arginine seems to be difficult for the methods to predict an arginine here. So none of the methods, either protein or ESM or variations, were able to predict an arginine here. But this contact is very important. So if you don't have this arginine, the energy is very worse than the wild type. Most of the methods predict some hydrophobic residue because you have a hydrophobic region here. So it's easier to the methods to predict a hydrophobic residue here. We are working now on some kind of multi-state design so that you can try to influence more this interface in contrast with the intrachain contacts. We try a lot of things, but the only thing that worked to get a design with a similar bind energy was to fix this arginine here and trying to identify other positions that contributed negatively to the energy and design them with ligand and pinyin. By doing this, we obtained design with better, maybe very close to the wild type. Another interesting case is the 7PB. Same case. Protein pinyin or ESM was not able to design something with energy close to the wild type. What we did, we're focused on only CDR3, that part that interacts directly with the peptide. But this time, we tried to expand the design to all the CDRs. So now we have all the interface being designed. By doing this, we found a design like this one here, I think. Yeah, or this one, I don't remember. That, I think this one. That showed better energy than the wild type. The explanation for this is that in the native structure, in a CDR, not the CDR3, in another CDR, I think it's the CDR1, there is this repulsive interaction between two arginines. And ESM IF1 predict a glutamic acid here, improving the interaction, as you can see in the composition of energies for this position. Yeah. Okay, we talk a lot about specific methods for protein design, using diffusion, graph-based approaches. But what about the LLMs, right? We are using now LLMs for everything, right? People ask, why not use LLMs for this kind of task? For instance, designing a new protein. I think last year, this article came out, and we have now maybe this question: could human AI contributions be the future of the interdisciplinary studies? In this study, they proposed to create a virtual lab. So the virtual lab is basically multiple LLM agents that talk with each other. They discuss the strategy for the design. They discuss the strategy for design new antibody for SARS-CoV-2, actually a nanobody. They can make decisions, they can solve problems, they can interact with external environments, they can execute actions, they can reason using available tools. They can run AlphaFold, they can run ESM, protein PNN, they can create a pipeline. So these are the phases of the project of design a nanobody to the SARS-CoV-2. You have, for instance, in phase 1, the human. The models will not like create the idea. Maybe they can, but in this case, they will not create the idea. Oh, let's design a nanobody against SARS-CoV-2. No, the human will do this. But you have other agents, like a principal investigator, an immunologist, ML specialist, a computational biologist, a scientific critic, that based on the input of the human, let's try to design those nanobodies, they talk with each other in meetings for selecting tools. For instance, I think it's better to use ESM. Maybe let's try to use AlphaFold multimer. Then let's go to Rosetta to see compute energy and see if your designs are good or not. The group can create the pipeline. They can access, actually, one limitation of those kind of models is that sometimes, this is basically, is based on ChatGPT-4, but sometimes the LLM are not updated. So you can, if you want, you can add more information, external information, with, for instance, more recently, the HAGs, retrieval augmented generation. And yeah, they can create the workflow and perform the experiments, not the in silico experiments. I'm not sure here, but they created some designs. They tested. Most of them are stables, but the success rate for the binding was not too high, right? So there is a huge room for improvement. Okay, and what's next? Next month, maybe some of them already read this, but people are commenting about the limitation now of AlphaFold, that AlphaFold is running out of data, and companies are trying to create our own data set to train those models, right? They are probably using OpenFold. So there is how can I say this? Not organization, but there is now a group of companies that the objective is to access OpenFold, which is open source, to train new models with private data sets. So each company has your own data set. Most of the data sets are focused on small molecules, right? Because we don't see a lot of PDBs with small molecules, and probably this is a limitation for training those models. So if you can generate more data, you probably can improve the pipeline for drug discovery. If you are interested in learning more about protein design, I recommend this GitHub. It's updated weekly, yeah. And it's very nice. Yeah, just to mention, we established a new protein design group in CNPEM. So If you go to Campinas, please visit us. You have a small symposium that we broadcast online on July. We bring Brian Pierce that works with protein design for vaccines and TCRs, antibodies. Also, Roberto Lins that works in a similar field. Leticia that works with design, engineering of enzymes. Yeah, that's it. Thank you. Any questions? Yeah. I'm not. How what? Yeah. Yeah. RF fusion can generate high diversity in terms of secondary structure and tertiary structure. But in my experience, it must generate helical structures. So unless you have some conditional to make it generate these other structures, it will probably not generate these other structures. Especially because you don't see this too much in the trained data set. Other questions? Yeah. Yeah. He's asking if I try to design residues that are not in the, for instance, in the TCR case, that are not in the interface, but that are in other regions of the TCR, right? No, I didn't, but in other projects, you try to do this because you know that you can, for instance, in antibodies, you can increase the stability of the antibody, trying to design, for instance, the core of the protein, and this is make some consequence in the affinity. We don't need to design the interface to increase the affinity. You can redesign, for instance, the core and increase the stability of your protein. But yeah. Other questions? No? Any announcements? Oh, sorry, a question?
The things you just mentioned.
Yeah.
Can they also be used in proteins that have certain allosteric functionality?
Yeah, he's asking if the tools that we use can design proteins with allosteric functions, right? I know that enzymes has a lot to do with this kind of allosteric, but no, we didn't. But and especially because it's not so trivial to understand the principles of the allosterism. So no, I'm not sure if people are trying to use this for design, but we haven't. Yeah.
To predict allosteric communications. So there are these efforts with variational encoder. So like Elder said before, there is a encoder, decoder. In the middle of the space, they extract the information correlate between residues, the motion, and everything else. So they can predict this communication between residues.
Thank you. Any other questions? No? So that's it.